My name is David Prescott. I'm a research scientist, immunotoxicologist at Health Canada in Ottawa, Ontario. And I'd like to welcome you guys to our afternoon session. And this one's it's a very food-related session. And I'm really excited about the speakers that we're going to have today. First, a couple of quick housekeeping notes. In this session, we're going to do the questions a little bit different. We're going to go the traditional way. So every time the speaker is done, there's going to be a little period of time at the end of the presentation where we can ask each individual speaker their a personal, not a personal question, a question that's, you know what I mean. Yeah, you get it. You're welcome to ask a personal question. They just might not answer you. Yeah. And then to start off, because my co-chair, Chio Saturn from Blue Nalo Foods, we're both safety minded. So the talks that we have are a bit about technology and a bit about the perceptions that go along with these technologies. And we're all going to be looking at ways of producing food in the future in places that it might have been hard to produce food in the past. And it's going to be fantastic. But first, we're going to do a little bit, I guess, of like a safety demonstration, like we're riding an airplane before the flight starts. Also, as a reminder, you can find the speaker's bio and the QR code, which is behind a sign right now. So if that doesn't work, I'm sure there are other ways that we can find it. There we go, the QR code. So next, if we can just jump into my slides, I'll get started right away with just a brief talk about the safety of novel methods of producing food. So I work at Health Canada. I'm a research scientist, and one of the goals that we have is to ensure that the food supply and the food that we consume is safe. And as a research scientist, sometimes my job is to look at exploring areas that have not been explored before. Thankfully, in terms of novel food preparation techniques and novel foods themselves, the government of Canada has a system in place to ensure that any foods that have not been consumed in the past will be safe for future consumers. So I'm just going to briefly touch on that. And this is the same for many countries around the world. I know the US is a little bit different. I'm going to speak from Health Canada's perspective. And a lot of the presentations earlier this morning kind of touched on how things work in the United States. But in Canada, we have something that's called the Novel Foods Program. So anytime that you have a food that I don't know how well you guys can see this here, but there are 3 different attributes that can get your food declared a novel food that requires it to go through this program in order to be approved for sale and for consumption in Canada. The first is a substance that does not have a history of safe use as food. The second, which has importance for this session, is any food that has been manufactured, prepared, preserved, or packaged by a process that has not been previously applied to that particular food. And also that causes this change also causes the food to undergo a major change. And the third, which is really the reason why Canada brought this legislation in the 1990s, is any food derived from a plant, animal, or microorganism that has been genetically modified to alter its characteristics. So this was really brought in with the dawn of a lot of GMO crops, and it's a way of the Canadian government to ensure that GMO foods would be safe to consume. So if your food meets any of these 3 criteria, you have to do the person or company that is making the application will have to have their food assessed for a number of different safety aspects. You see them listed there. I'm not going to go through them all, but you have to ensure that the food is safe and good for consumption throughout all of those different attributes. And each applicant will then have to make a case-by-case application, which are not prescriptive, which means that there's not a set of experiments or a set of assays that you have to do. As long as you satisfy the reviewers that your product is safe in each of these aspects, you can do it kind of however you want. You can do it either by literature, read across, you can do it by the assays themselves. But this is a way of ensuring that something that has never been consumed before meets all of these criteria before it can be purchased and eaten in Canada. So we'll see how this, some of these things may apply to some of the talks that we have later. But in my world, my main focus that applies to this is cultivated meat, or sometimes referred to in popular media and others as lab-grown meat. So meat that's grown in a petri dish. And if you think about it in these 3 different definitions here, lab-grown meat or cultivated meat could fit into sort of any of these. But seeing as we're focusing on new production techniques, lab-grown meat is definitely something that has been eaten before. We have eaten, obviously, muscle tissue from animals. This is just muscle tissue that has been produced in a way that has never been produced before. So one of the aspects of this production that is new, which makes it novel, is the presence of culture medium. So when I think of culture medium, I'm a bench scientist, so I think of like the little red bottles you get from Gibco that look delicious, but I've never tasted them. I don't know. I mean, I don't. I imagine they probably taste very salty and terrible as well. But I never really think much about what goes into it. When you break it down, a lot of the things that are in culture medium are things that you would find in food, like vitamins and some salts. But there are some things that, such as the components that are used to replace fetal calf serum, fetal bovine serum, are things that are not necessarily used often in food. So it's important that when we're assessing the safety of cultivated meat, we think about the ingredients that are in there and the level of exposure. So I didn't add all of this stuff about risk versus exposure afterwards frantically at lunchtime. This was actually in my talk before. It's important to note that the level of exposure from the culture medium in the final product isn't necessarily going to be the concentration of the ingredient in question in the product itself. So as a research scientist, one of my jobs is also to figure out, do we have enough information to confidently assess risk for each of these ingredients? So this leads me to the main sort of reason that I'm speaking about this, is the pitch that I'm making for the work that we're doing here at ILSI in this very space. So this leadership committee for cultivated meat over on the right, there are a number of these people are in the room today, or at least a few of us are in the room today. But together we're working on an exercise where we take each component of a model culture medium and do a little mini risk assessment of each one. And at the end, we can then assign each of these components a risk tier that's based on some of the guidance from the FDA. So this is the language that we have to describe which tier of risk each of these components fit into is a little bit more comprehensive than what I have listed on the screen. But the main take-home message is that the tier 1 ingredients are approved for use in food without restriction. So the vitamins that don't have any upper level limits. Tier 2 are things that are approved for specific uses in food, but have well-characterized upper limits. So, obviously you don't want to go above these upper limits, which might include some other vitamins that do have upper limits. There are vitamins, there are daily upper tolerable limits. Tier 3 are things that are ingredients that don't have any regulatory status, but are expected to be found naturally in food. So there's a lot of things like putrescine or some of the other components that are found in culture medium and in fetal bovine serum that aren't used as a food additive, but you would expect that any time that you ate fruit or vegetables or meat, you'd be consuming probably much greater levels of putrescine than you would be finding in cultivated meat. And then tier 4 are the ones that have no regulatory status and are expected to be found, are not expected to be found in food. And these are the ones that are probably, we're going to require the most regulatory attention. So what we have is, and this is for a vitamin B5, kind of an example of what the group has put together by going through each of these, and we've all sort of split this up into individual ingredients and tackle the problem collectively. But we get a little summary like this, and I'm currently in the process of compiling these all into a text document that I'm hoping that we'll have. It's really, when I say I'm hoping, it's all up to me. So this is a reminder to myself to get going. Later in the year to have this published someplace. There'll be the goals of this are multiple. So it's useful for regulators to be able to look through and know the ingredients of culture medium and sort of have this risk assessment done for them already. It's good for companies who are making submitting assessments, because then they're able to look and use this as an example of how you can do it and how you can organize it nicely. And also the ranking exercise is going to be interesting because we're going to be ranking each of these things individually, and it might give us some indication, no matter how well defined we think the tiers are, there might be some discrepancies. And that's, I think, an important characteristic that might come out of it. We might find out that half the people think an ingredient is tier 3 versus tier 4, proving that it's not necessarily, there is still some subjectivity to it that we need to tease out before we can start really using this as a regulatory method. So that's my little pitch for our thing. And now I'd like to welcome up my co-chair.